Hi, and welcome to this video where we're going to be running through 20 multiple choice questions on the topic of disease and pathogens and immunity. So that's module 4.1 from the OCR A-level biology syllabus. Now again, weirdly, the exam board haven't published anything, so these are some questions that I've cobbled together. So if you're one of my students, then what you should do is you should first do these questions, and then you should watch this video to mark them. If you haven't got access to the file of these questions, then what I suggest you do is just pause the video. We'll be going through sort of about 3 questions at a time, so pause the video when you see the question, do it, and then see how we go through it. Okay, so number 1, let's get started. Tuberculosis is an infectious disease that affects humans. It is caused by a pathogen. Pathogens can also cause disease in plants. Which of the following plant diseases was caused by the same type of pathogen? Okay, so tuberculosis is caused by bacteria, so that's the first thing you need to know. So which plant disease is also caused by bacteria? And this one is ring rot, okay, that one, C. This is caused by a fungus, this one. Mosaic, that's a tobacco mosaic virus, V for virus. Late blight is actually a protist. So the answer is C. Okay, on to question 2. Which of the following descriptions is correct? Now, this is a bit of a tricky one. It's quite sort of wordy, and it's kind of looking at the difference between vaccination and immunization. So I'm just going to go straight for the best answer here. This is the answer. Double check with the exam board because this one caused me a bit of confusion. Vaccination involves injection of antigenic material, and immunization is the process of developing immunity. Yeah, so that's the answer. Typically, when we're sort of speaking a bit more casually, the two terms are used interchangeably, which is what can cause a bit of confusion. But the answer from the exam board is B. Okay, on to 3, last one on this page. Which of the following formula would you use to estimate the volume of a neutrophil? Well, a neutrophil is roughly it's circular. It's roughly a sphere. So which one is the volume of a sphere? It's this one, 4/3 pi r cubed. So the answer is D. Okay, moving on to the next set of questions. Zoom out a little for those of you who don't have the questions. There we go. Okay, so 4 and question 5. Do those now if you need to. Let's continue. So, which of the following numbered parts of the diagram represent the part of the antibody that has the same sequence of amino acids in all the antibodies? Well, this is the constant region, isn't it? So it's this one. So it is this part. Now, this is kind of tricky to say where this is pointing to, this one. This definitely has the same region. This up here is the variable region. So this is a bit of a tricky one because it is 2. What's 2 pointing to? Is it pointing to this specific part or is it pointing to the whole chain? I think it's pointing to this specific part, in which case that is also part of the constant region. So I'm going to say that 1 or 2, 1 and 2, sorry, 2 and 3, 2 and 3. 2 and 3 are the constant region and they have the same sequence of amino acids. This is the variable bit up here, the bit that balances the antigen, so that's the one that has different amino acid sequence. Okay, moving on to question 5 there. Which statement A to D correctly describes a process that provides artificial active immunity? So artificial active immunity is active immunity, first of all. That means you're making your own antibodies. And artificial means it's a vaccination, basically. So an injection of active antibodies, look, that's a red herring. It's trying to say active antibodies. No, if they're injected into you, that doesn't mean it's active immunity because that's a passive process. You're just receiving the antibodies. It's this one. Okay, antigens for polio given in a sugar cube. This is a tricky, so this is a type of vaccination. So for polio, the vaccination is actually an oral vaccination. Basically, there's antigens on the sugar cube. You eat the sugar cube, your body learns to recognize the antigens from the surface of the virus and generates your own antibodies. Your body is actively making your own antibodies, but it's been artificially trained to do so. Okay, all right, moving on to the next few questions. 6 and 7. If you need to do those questions, pause the video now, do those too. Okay, all right, 6. A patient has been diagnosed with an autoimmune disease. Which of the treatments A to D is most likely to relieve the symptoms? So first of all, remember autoimmune is when the immune system attacks yourself. It attacks your own body, tissues or cells. So how are we going to stop that? Antibiotics, no, that will only work on bacteria. This is just weird. A vaccination containing antibodies, well, A, that's not a vaccination, and B, that's not going to do anything, so no. Drugs to suppress the immune system, yes. Your immune system is attacking yourself, you want to calm it down, so that's going to be the one. No, surgery, that's pretty drastic. So 6. The answer is C. 7. Here's another one about what type of pathogen causes what type of disease. So which ones of these are correct? Athlete's foot and ringworm are caused by fungi. That is correct. Both of those two are caused by fungi. Malaria and tuberculosis are caused by protoxins. What do you think? Incorrect. Malaria is, tuberculosis is caused by bacteria. So that's a little hidden incorrect thing in the middle there to try and throw you off. Ring rot and black sigillococca are caused by bacteria. That is also incorrect because this one is caused by bacteria, but black sigillococca is caused by fungus. So the only one that is correct is 1. So D is the correct answer. Moving on. Okay, let's look at, I'll just zoom out. Well, let's do them one at a time. Okay, so question 8. First of all, if you need to do it, have a think, pause, and we'll come back to it. Okay, right, the graphs show the concentration of antibodies in the blood of 4 people after a first natural exposure to an antigen. One of the people had been vaccinated against this antigen previously. Which person has been vaccinated? Hopefully this one should be fairly clear. The answer is going to be B, because look at this quick increase in the antibody concentration in the blood. This is due to the presence of B and T memory cells that are sort of present at T0 and rapidly clone themselves, clonal expansion, and we get quick secondary immune response there. Okay, on to 9 about lupus. Pause the video if you need to do it. Okay, so it's an autoimmune disease. You can get a facial rash, typical on a butterfly shape across the cheeks. Following a blood test, which of the following would indicate a patient has lupus? Zoom in a little. So, is it the presence of antibodies for the cell surface antigens of connective tissue? Now let's think about that. So you're producing antibodies against your own tissue. So yes, that sounds right. But let's just put a little question mark and keep going. Presence of herpes antibodies. No, that's just showing that you've had herpes. The presence of high levels of antihistamines. That's more to do with allergy, like if you're suffering from hay fever, so probably not, but we'll put a kind of potential. Absence of B lymphocytes, well, that's some sort of immune disorder, so no. The best one is A, which is the correct answer. All right, moving on. Let's pinch out a little so you can do these 2, 10 and 11 if you haven't got access to the file. Pause now if you need to do them. And now we're going to get started. Okay, 10. An individual bitten by a rabid dog. The treatment is actually you get an injection of antibodies, human rabies antibodies. So what type of immunity is this? Well, it's artificial because you're having an injection. So it's definitely one of these two. And is it active or passive? Remember, active is only if you are making your own antibodies. So this is passive because it's an injection of ready formed antibodies, 10C. Okay, so here's a blood smear. What is visible? Okay, right. Okay, so we've got here RBCs. This is probably something you should have done anyway, just try and work out what's what. That's going to be a neutrophil because it's got a multi-lobe nucleus. I think that's a neutrophil, and I think that's a neutrophil. I think they're all neutrophils. This here could be a platelet. I'm not sure. But there's no platelets here anyway, so I don't see lymphocytes because remember, Lymphocytes would be, let's just try and keep the same color scheme. Let's go and draw it in dark gray. Lymphocyte would be, you know, something that's like this, but then it would have a very large nucleus that takes up almost all of the space. Something like that would be a lymphocyte. So lymphocytes are definitely not there. So it's definitely one of these two. The erythrocyte's definitely there. One of those three. Neutrophil's definitely there. So the question comes down to whether this or this is a monocyte. I think no. So I'm going with 11B. Am I right? Yes, it is right. So a monocyte was a bit hard to tell. A monocyte is a cell and it has a large kidney-shaped nucleus, kind of a bit like that is a monocyte. Now, this is not an accurate biological drawing. I'm just kind of showing you roughly what it looks like. Okay, so that's a monocyte there. A monocyte is something that can become a macrophage when it moves out into the tissues. Okay, moving on. 12, 13, and 12 and 13. Okay, pause if you need to do these two questions. And let's continue. Which of the following options, A to D, is a primary defense against pathogens? I had to look this up. I remember this when I did this earlier, because I don't really like this, but okay, it kind of bugged me, this definition. Okay, so primary defenses, remember the definition for primary defenses are things that stop the pathogen from entering the body. And I'll tell you why this bugs me. Antibody production, no. Okay, because that only helps against things once they're already in the body. Okay, let's skip this one. It's this one. Okay, this is the answer. But let me tell you why it bugs me in a sec. Phagocytosis, no, because that's one thing that get in the body. That's a secondary response with white blood cells. Once they detect pathogen in the body, they try and eat them. T killer cells, that's way down the line. That's part of the specific immune response. So that's quite far down the line. Definitely not primary. Inflammation can be considered to be primary, even though it generally happens when you have a cut. So something's already in the body and then it gets inflamed. I guess the reason why is inflammation the role of it is it stops spreading from a local area. So if you get a cut and bacteria get into that local area, it becomes inflamed. So it tries to stop the spread of the pathogen from the cut out, and it does that by making the blood vessels leak, so that you kind of have sort of fluid is trapping the bacteria in that area and not allowing it to sort of flow away. You have blood clotting responses. You have neutrophils moving out of the capillaries to target the area. So it bugs me because I feel like it hasn't stopped the pathogen entering the body. But I guess in terms of the definition that the textbook uses and the syllabus uses, it is a primary defense because it stops it spreading from a localized area. So that's the answer. I don't really like it that much, but that is the answer the exam board wants. 13. 3 methods of pathogen transmission between animals or plants are listed below. What can apply to plants? Okay, contact. Yeah, a plant can contact another plant. Maybe there's an infection in a leaf and wind blows the plant and it blows over here and touches this plant, and then this plant has the infection and it can blow over and touch the next one. So yeah, vectors. Yes, definitely vectors. So what might be a vector for a plant? Well, maybe something like an aphid could be, or a beetle. Something like that. An aphid goes and feeds on one plant, picks up a virus with its mouth parts, goes and feeds on another plant. And then we have a vector transmission. Droplets. No. Okay, so plants don't cough and sneeze and they don't breathe. So therefore, we don't have any droplets knocking about. So it's only 1 and 2. B. Okay, 14. If you need to pause, do so now. Which of the following types of cells is not involved in a primary immune response? Okay, so this we're not talking about primary defenses and secondary defenses here. This is primary immune response and secondary immune response. What we're talking about is this graph where it kind of goes like, whoa, whoa, like that, okay? So this is the primary response, and this is the secondary response, and this is antibody concentration over here, and this is time after first infection here, and then after second infection here. So, yeah, these ones, they're not involved. Memory cells. There is no memory of the pathogen if it's the first time you've met it. Helpers are involved, killers are involved, plasma cells are involved, so the answer is A. 15 and 16. Pause the video if you need to do these two questions. And now let's get going. Okay, so plants such as the soybean have a bunch of different defense strategies, which is a chemical defense against pathogen infection. Okay, right. Which is a chemical defense? Ooh, okay. Callose deposits at hydrolytic enzymes such as chitinase found between cells. Stomata can be placed. This is okay. This is definitely not. That's physical. Difficult entry. I'd say that's physical. Yeah, that's sort of more physical. Okay, so the best one. I had to think about that one actually a bit. Hydrolytic enzymes. Okay, that's the only one that is involving a chemical reaction, an enzyme causing the breakdown of, for example, fungus. Okay, fungus contains chitin, so hydrolytic enzymes would break down the fungus if it tries to grow through the plant. So 15B is the best one. That's a little bit of a tricky one. All right, 16. Which of the following statements about antibiotic resistance is correct? No, okay. All antibiotics cause mutations in bacterial DNA. No, they don't cause them. It's a selective pressure thing. Antibiotic resistance in bacteria is evidence to, yeah, that's true. Development antibiotic resistance in bacteria is an example of genetic drift. This is a term that we will use in year 13 when we're talking about speciation and sort of changes in a population, but this is not the right time to use this term. Genetic drift is random change in a gene pool just due to random mutations without any real selective pressure. Again, this is another red herring. This is the year 13 terminology, so you wouldn't have heard it yet. This is something we cover in year 13, and it refers to when there's natural selection at two extremes of a population, let's say, so that very short animals and very tall animals don't survive, and only the animals in the intermediate height survive, and then that's called stabilizing selection. We'll look at that in year 13 then. All right, moving on. 17. Do the question if you need to. And now the answers. Okay, what increases the phagocytosis of pathogens? Yeah, opsonins. Remember, definitely. Opsonization is when the antibody sticks to a pathogen, and then the back end, the constant region of the antibody, this is an antibody here, this is the back end, this bit has a signaling molecule that kind of signals to the white blood cell. Agglutinins, antitoxins certainly not. Agglutinins, I think so. I think if you agglutinate multiple pathogens together, it makes them more easy to kind of take in. So I think that it is 17B, and I'm checking, and the exam board agrees with me. Yes, it is. So when you clump pathogens together, so imagine these are bacteria here, and they're all clumped together with some antibodies that are kind of sticking them all together like this, like that. They're all clumped, and then along comes a white blood cell here and does its whole phagocytosis thing. That's a nucleus, by the way. Let's make it a neutrophil. Multi-lobe nucleus. Yeah, okay. So yeah, moving on. Question 18. Okay, what's this? Hopefully you recognize it straight away. Pause if you need to do it. This is a virus. Okay, we should be pretty familiar with what a virus looks like. We've seen countless graphics of coronavirus on the news for weeks and weeks and weeks. So this is the one. Now this type of virus is a retrovirus. Okay, so we didn't do too much on this in the mind map, but a retrovirus, what it does is it has RNA and it carries this special enzyme here, reverse transcriptase, so that enzyme, if you think about it, it's reversing transcription. So it takes RNA and turns it into DNA, which can then be, there's another actual enzyme in here which isn't labeled. Oh, there it is. Look, that one. That one is actually called integrase. And what it does is the integrase actually puts the DNA into a chromosome in the nucleus, which is where it stays, which is why HIV is so difficult to treat. because it's basically DNA edits itself into your genome, and then it's hard to get rid of. Okay, moving on. 19 and 20. 2 questions on a similar thing. Okay, pause if you need to do this now. All right, percentage increase in the number of cases from 1995 to 2000. Okay, so 1995 to 2000. First of all, let's identify the numbers. So 1995, we've got 10,000. In 2000, I think that's 65,000. Okay, so first of all, what is percentage change? It equals to the difference, or the change, over the original starting value times 100 to make it percent. So the difference from 10 to 65 is 55,000 over the original, which is 10,000 times 100%. And can I do that in my head? It's going to be a big number. Is it 550 or 555,000? I've got to do this on my calculator. My brain's not very fired up yet this morning. Divided by 10,000 equals times 100 equals is 550. Okay. B. The next question follows on from the same thing. Okay. So here it is again, same graph, but now it's asking us which of the following statements could explain the data from 1999 to 2000. Okay, so that's this actually just this year. Okay, just this bit here from 1999 to 2000. Pause if you need to do it. Okay. All right. The malarial parasite had developed resistance to insecticides. Well, okay, no, because malarial parasite itself, that's not an insect. Maybe mosquitoes are insects, and maybe they developed resistance to insecticide, but not the malarial parasite. The vector had developed resistance to antibiotics? No, because the vector is mosquito, so that doesn't make any sense. The malarial parasite had, no, okay, that doesn't make any sense either because it's not a virus, it's a protist. The vector had, yeah, there we go. The vector had developed resistance to insecticides, yeah. Okay, so that's D. Okay, so that's the end of that video on pathogens. So I hope you enjoyed that. Make some notes if you've got any questions wrong. You can add it to your mind map, for example, and you can review your textbook if you felt you needed to do a bit more work on any certain area. Okay, thanks very much, and I'll see you next lesson.